Welcome to From Our Neurons to Yours from the Wu-Tsai Neurosciences Institute at Stanford University, bringing you, as always, to the frontiers of neuroscience. We probably all know stories of an older friend or a relative who went into the hospital for a routine surgery or something more major, like a hip surgery or cardiac bypass. And when they came out, they just weren't quite the same. Something had changed in their thinking, their memory, but the story is all too common. The question is this just anecdotal, or is there something more important going on here? It turns out this is something that surgeons and neurologists have been aware of for a long time, but there's not actually a lot of great data that links major surgeries to people's vulnerability to cognitive decline and that slide into dementia that sometimes happens all too suddenly. Today's guest, Martin Angst, is an anesthesiologist at Stanford who is trying to turn this question on its head. In his work with the Knight Initiative for Brain Resilience, he's asking not just what predicts whether someone is at risk of cognitive decline after surgery, but why is it that some people bounce back even after a major invasive surgery like a cardiac bypass? This is something that really ought to affect the brain, but some people seem much less affected. Why is that? Martin's interest as an anesthesiologist in how we heal after surgery has led to a major research study that he's conducting with the Knight Initiative, following hundreds of patients both before and after major cardiac surgery, looking at all kinds of factors from blood biomarkers to cognitive performance to try to understand what are the factors that predict resilience or vulnerability to cognitive decline after surgery. In our conversation today, we'll explore what Martin and his team have discovered so far and what it might mean not just for how we help advise people going in for surgery, but also for our understanding of brain resilience more broadly. Let's get into the conversation. Martin, welcome to From Our Neurons to Yours. It's so great to have you on the show.
Thank you. Thanks for having me.
Martin, the first thing I wanted to ask you about, we've been putting together this article for the Knight Initiative for Brain Resilience about the work you've been doing. And there was a moment in it that stuck out to me. I know that you're a clinician scientist, you're an anesthesiologist, and you also have a research lab. And my understanding is that your research started out focusing on sort of the mechanisms by which opioid drugs work in the brain, but that about 10 years ago, you sort of had a change in the focus of your work. You switched up what you were interested in studying. I wonder if you could think back to what was going on at that time and tell us a little bit about how you came to the area of research that you're focused on now.
Yeah, so there was definitely a reflection point in my scholarly endeavors at Stanford. As you pointed out, I started out in clinical pharmacology, and I had a significant interest in understanding opiate pharmacology and the benefits, but also the potential adverse effects of opiates. Many listeners will be familiar with, obviously, the opiate epidemic crisis, addiction. Yeah. We studied some of the biology, and in the early 2000s, there were these studies that we sort of looked at candidate genes, and we were sort of interested in understanding some of the genetics and the genetic background, and if you could explain why some respond favorably and some respond non-favorably to opiate pharmacology or opiate exposure. That sort of came to a halt because as technology advanced, whole genome sequencing became a big thing, and you need thousands of participants to study the genetics in a meaningful way. And it became clear that what we could do in the lab setting at Stanford became quite limited, given these new technologies. At the same time, it did strike me that as an anesthesiologist, I witnessed patients undergoing surgery every day, and very close to what I do as a clinician was the fact that these patients all sustain significant bodily injury. Obviously, the intention is a good one, right? You want to basically replace a failing hip or knee, but the way to do this by surgery goes along with a bodily injury.
The surgery itself is the major injury.
Yeah, surgery itself. And so it did strike me that there was a significant opportunity to actually study the biology of healing from injury, and that the operating room in some ways is almost like a laboratory. where you could study human biology, it was very close to who I am as a clinician and what I witnessed. And so the limitations in my sort of first life as a researcher at Stanford and the opportunity I had that just did strike me as this is really what I should do. And so that's how it all started.
I love that idea of using surgery as an opportunity to study healing. We talk sometimes about natural experiments. This is maybe not a natural experiment exactly, but a necessary experiment. Someone needs cardiac surgery, someone needs a hip replacement. They need it. They really need it. But in the course of that, there's this whole injury and healing process that goes on. And I love that perspective as an anesthesiologist. I mean, we've had Boris Heifetz on the show from your department, and sort of the perspective of the anesthesiologist as a part of the clinical team who's really responsible for the patient's well-being and recovery in a certain way, aside from the clinical procedure that's actually going on, is quite interesting to me.
Yeah, in many ways, you could think of us as travel companions. We think about patients ahead of surgery or ahead of injury. Can we optimize how they come to surgery? Can we basically make them fit for surgery? Are there things we can do so they will basically recover faster? So we are companions ahead, and then obviously we are there with the patient during surgery, and then we think about how do they recover, how do they heal. So there's this continuity of care that may not necessarily be obvious if somebody thinks about an anesthesiologist. And in many ways, we think about patients on a systems basis, right? It's not an organ that we think about like a heart or a liver. All of these organs, the patient and all the humanity around the patient, all of these things act together. And as anesthesiologists, we ideally think of all these things. How do you come in? How do we get you through? How do you recover? How do we basically transition you back to a normal life or hopefully a better life?
Yeah. So as part of this new focus of your work, you took up a collaboration with Igor Feinstein and Michael Gracious, who's a friend of the show. He's been on the show before, and other colleagues at Stanford to try to understand this somewhat anecdotal observation at the time that often after surgery, particularly for people who are older in their 60s, 70s, 80s, there are often these stories that after surgery, people experience some level of cognitive decline, problems with memory, with thinking, recognizing people, and so on. How well was that known or documented at the time that you and your collaborators started this study?
I mean, as you pointed out, there are a lot of anecdotes or patient reports. The story typically goes, I had surgery, a hip replacement. My dad, my mom had a hip replacement. And, suddenly they couldn't recall names, phone numbers. And to your question, how much do we know about this? We do know that surgery, and particularly major surgery, including cardiac surgery, is associated with what we would call post-op cognitive decline, or let's say impaired brain health. You mentioned memory function, which is the most obvious one. So that's documented. It's a low percentage of patients that will suffer permanently from cognitive decline. What's also not well understood is why is there so much diversity? So some patients seem to do just fine. They have the same surgery, the same insult, the same age. They may even look quite similarly clinically, but then others basically suffer from these consequences. And so in today's world, given many of the technological advances that we have, we can now get into the question, what are the biological determinants next to clinical determinants or epidemiological determinants that actually drive cognitive health or cognitive decline in the context of suffering from a major bodily injury? Cardiac surgery is one example.
Well, I love that you're sort of highlighting two sides of the coin there, right? Which is we see that many people have this experience of either temporary or in rare cases, permanent cognitive decline. Why does that happen? What's going on? That's sort of I feel like, typical response of clinicians and scientists, like, here's a problem, what's causing it? But then flipping it all over and saying, well, but it doesn't happen to everyone. There are a lot of people who are perfectly fine. Why are they fine? We're doing this major intervention. We're cutting open the body. We're messing with their organs. We're rerouting their bloodstream in the case of cardiac bypass. How is it that they're fine?
Yes.
So tell us a little bit about the genesis of this 2021 study that you published. What were you trying to figure out specifically in that study and how you set that up?
Yes, this study was initiated based on actually a particular interest of Igor, who is helping conducting the current study. And Igor and I have a mentor-mentee relationship. We have known each other for a long time. And he was particularly interested as a cardiac anesthesiologist in cardiac anesthesia and consequences thereof. One of his major interests was related to understanding how the brain responds. He was particularly interested in imaging technology at the time, and we designed a study using imaging modalities to sort of look at brains ahead and after surgery. It turned out that this was a huge challenge and it became impossible because COVID hit, and we were basically sitting on multiple patient samples as we collected serial blood from these patients enrolled in this imaging efforts. And that's when we met with Mike. Mike said, you have these wonderful samples and you have this major surgery, as you can think of it as a stress or an intervention. Let's look at these samples and let's look at some of the biological markers that we sort of know are associated with brain stress or brain insult. And we said, we have these samples. The study came to a halt. Let's look at these samples. And we looked at some proteins that are associated with neuronal stress and neuronal death. And we got the results back, and we were all a little shocked by how dramatically these proteins became elevated during and after surgery. So there was a protein that relates to neuronal stress. So you would think if that protein is elevated, neurons in the brain have been stressed. And we observed 6 to 10-fold increases. Wow. Again, importantly, not everybody, but some patients. And then there is another protein that became elevated after surgery, and that protein is related to neuronal death. So an indication that actually neurons die. And again, 6, 7, 8-fold elevation in some patients, but not in others. So that was how this all started. We said, wow, these patients undergo this intervention. And we have now markers in blood that tell us something about are neurons stressed, and subsequently may neurons actually die.
And these were in patients both undergoing a cardiac bypass, and then other patients were having a hip replacement, if I remember right? Is that right?
Yeah, that's we had these two populations, and interestingly enough, hip replacement surgery, was associated with similar changes, but they were less pronounced than what we observed in patients undergoing cardiac surgery. And that likely relates just to the type of injury. Cardiac surgery is probably one of the most invasive procedures, and for good reasons. But the fact that we saw quite dramatic differences between these two populations indicated that the type of surgery and the type of injury and the magnitude of injury actually matters in how relevant this becomes.
And I appreciate you talking about this at a high level and not dipping us in the pool of jargon. But in this case, I mean, these are also proteins that are associated with things like Alzheimer's disease, right? I mean, we see the same protein, I think it was phosphorylated tau and neurofilament light. I mean, this is word salad for some people, I'm sure, but these are proteins that experts in Alzheimer's disease would immediately go, oh, that's not good. We don't want to see that in the brain.
Yes. You see that in plasma, and you talk about the p-taus. The p-taus measured in plasma or in blood are highly predictive of Alzheimer's disease years down the line. So these markers become elevated years ahead of anybody becoming clinically symptomatic. So p-taus, NFL, neurofilament light, those are markers well known in the field of cognitive decline, including Alzheimer's disease. So that obviously was another connector where we all go like, that is concerning.
Yeah, very concerning. Well, I mean, the thing that it speaks to me, and I think you mentioned this earlier when we were talking about sort of the anecdotal reports about everyone knows someone who went in for surgery and came out and it took them a while to get back to their baseline. I mean, it makes me think of these studies that have been coming out recently about how aging is really nonlinear, right? It's not, we don't age the same amount all the time. We'll often see with relatives and, sometimes looking in the mirror, aging can happen very fast at times. And so this opportunity to look at what is one of these stresses, one of these things that can really dramatically change someone's cognitive state is a really interesting opportunity.
Definitely how we look at it. And I also want to put, as concerning as it is, a positive spin on it. We now have the tools to study resilience. If somebody is really vulnerable, we may want to think about how invasive do we want to be going forward. And I can think of ways we could actually consult patients and down the line give them data-driven and biology-driven advice of you are in a high-risk group or you're not. And the other thing, and you mentioned that initially, is what are we going to do about it? And could we actually make patients more resilient and give them hope that we will fare well? And that's probably something we should talk about.
Absolutely. Yeah. So, Martin, I want to talk more about this idea of where do you go from here now that you've seen this elevation of these concerning molecules in the blood plasma of people undergoing surgery and in the weeks following. But before we do that, I feel like we need just a bit more of a picture of what does this surgery actually involve? What is actually going on when someone is going through a cardiac bypass surgery? You've mentioned that it's a significant challenge to the body and to the brain. And what do you think might be triggering some of these signs of neuronal death and stress?
Yeah, I mean, the patients we study in cardiac surgery are patients who go on bypass, which means the heart stopped. Because the heart is stopped, you need to oxygenate the blood. In order to oxygenate the blood, you have that blood diverted out of the body. You oxygenate the blood and then you sort of give it back. So you can think of it as, a hose coming out of the body, and then blood gets oxygenated and then blood is fed back to the body while the heart is arrested. Just to get to the heart, you open the chest. So you have significant incision, significant tissue that's dissected, but then also blood diverted to an oxygenation source outside the body. And all of these things cause inflammation. And inflammation has been recognized as a driver of adverse outcomes. We think that patients who inflame a lot are at higher risk. Now, obviously, you need some inflammation because inflammation is the way you initiate healing. You respond to insult and you initiate healing. But maybe there is too much inflammation. So the term we use clinically here will be you're a hyperinflamer, or you inflame particularly above what an average person would inflame in a given circumstance. So in many ways, cardiac surgery triggers a significant inflammatory response. This inflammatory response varies based on who you are biologically. And then we think the magnitude of how much you inflame is responsible for driving some of the outcomes. And we sort of know that from the trauma literature, that patients who suffer trauma and who inflame dramatically, as measured by blood tests, are patients who are more likely to stay longer in the ICU and are patients who are at higher risk of significant morbidity and actually mortality as well.
I mean, hearing your description of what is actually going on during cardiac surgery, I really get a better sense of what you're saying about how amazing it is that some people recuperate so well from a procedure like this. And, it's maybe not so surprising that there are some cognitive effects of something major like that that's going to trigger inflammation and involve a lot of healing. So after this study, if I understand right, I mean, this is when you and your collaborators partnered with the Knight Initiative for Brain Resilience, or got support from the Knight Initiative to look at a longer timeline. What is actually going on before surgery? What's going on after surgery, following these patients for a longer time. Can you tell me a little bit about the group of patients that you have brought together to go on this research journey with you?
Yeah, all these patients obviously have heart surgery using bypass, the methodology that I described to oxygenate the blood while the heart is in arrest to do the surgery. The patient population is typically 60 to maybe 85 years old, so it's an elderly population. We recruit patients from all over California. Patients are coming in with comorbidities. Obviously, they come in because they have a heart issue. Sometimes they have other health issues. So it's an elderly population undergoing major surgery, and some of these patients have significant other comorbidities.
And they're volunteering to have you take their blood and spinal taps as well?
We do if there is a catheter placed close to the spinal cord. That on its own is required in a minority of patients, but we do serial blood samples. So we study these patients longitudinally, so before surgery and then multiple times after surgery. And then we follow 3 months after surgery and then 1 year. And all of the patients have agreed to be part of the study for at least 5 years. Now, moving forward, we would love to study these patients lifelong and see how they evolve. We look at cognitive outcomes. So we have a battery of tests that we can apply remotely. We send them iPads and then we communicate them through iPads. And we test them in 14 or 15 different domains of what we call cognitive function, which includes memory functions, long-term memory, short-term memory. The idea being that we really follow them as long as we can. And that's a significant shortcoming of many of the studies that have been done in the field. Many of these studies end at 3 months or 1 year. We're really interested to understand what does that actually all mean long term, and then also related to things like, okay, so we have these test results. What does that actually mean for how you function in your life?
Yeah, I was going to ask, what is it sort of big picture that you're hoping to learn from this long-term study?
I mean, the big picture is we would love to learn from resilient people as to why they're resilient, and we would love to use that knowledge to help patients who are not as resilient or vulnerable. All of that has to be somehow relevant to the patients. Can you actually do the things you do in your life? Daily activities, be socially active, hang out with your friends, be back to a level that makes your life enjoyable. So that's our mandate. Research is exciting, but at the end, I'm also a clinician. And at the end, I'm also a human being. So
Right.
I'm really interested in, can we make our patients as well as they can be?
So I know you're only a couple years into this effort, or maybe only a year into this effort. Is there anything you can share with us so far about what you're starting to see with this patient population before and after surgery?
Yes, I'm happy to share our results. I mean, they are preliminary. We have now studied about 80 patients. We hope to study up to 500 patients long term. But the first thing that we learned is what we observed in our really small study initially happens to be true. So these p-taus peak dramatically during surgery, 6-8-fold in some patients, followed by peaks of neurofilament light. And neurofilament light, an indicator of neuronal death, actually continues raising until patients are discharged. It's not like day 1 or day 2, that's the peak. Actually, if you follow patients to day 7, these levels go up. We have been able to relate the magnitude of this elevation to some of the cognitive outcomes that we study, at least for 3 months. So we have strong indication that if you have high levels of neurofilament light after surgery, you're more likely to have memory issues 3 months out of surgery.
Just to make sure I'm tracking, so that you're seeing this p-tau indicator of neuronal stress during surgery.
Yes.
And then in the week following surgery, sort of until discharge, you're seeing this increase in neurofilament light, which is interpreted to mean cells are actually dying off, cells that got overstressed.
Yes.
And you're saying that connects to the level of cognitive decline up to 3 months later?
Yes. So the other thing I want to point out is the response, if you think about the biology, is drastically different in the patients we have studied. So again, there are patients who have really, really high levels, and there are patients who don't. And who are these patients? It's not all explained by the type of surgery. It's not all explained by the particular chronological age. So there is a predisposition to be resilient or predisposition to be vulnerable. Now, the question is, can we actually capture that? And at some point, you have, okay, John is showing up for cardiac surgery, and you can say, John, based on what we can measure, you very likely going to be resilient to the intervention, or you may be vulnerable. What does that mean for this patient's journey? If you think about informed consent, this could be the next level. A patient could actually make an informed decision and say, I understand the risk, but I'm willing to take the risk because there are benefits to this particular procedure. Or you could say, maybe we choose an alternative pathway, a pathway that's less invasive, less disturbing to the system. So there's the hope that we can make meaningful prediction that would eventually allow us to be competent consultants to our patients? Can we do something about making you from being vulnerable to making you being resilient? And what we learn in surgery, we may actually be able to translate into other settings. So what we learn here may be highly relevant to cognitive health in a population that doesn't necessarily undergo surgery. So hopefully what we learn here in terms of biology has broader implications. And I think that resonates well with the Night Initiative, which is not focused on surgery, but general brain health.
Right. I mean, it's maybe not completely surprising to listeners that with an intervention with an insult, as we say, as extreme as cardiac bypass, it's going to have cognitive effects. There's going to be healing required. But it's such an extreme thing that to see resilience, even to such an extreme intervention, makes it a really interesting model to say, okay, if you can be resilient to this, what's the secret of being resilient to something as extreme as cardiac bypass? And as you say, that's something that hopefully we can take and apply to other areas as we age, as we look at other ways that we can make our brains resilient to the insults of time.
Yeah, I like that spin. I think that's really it. That's the inspiration here.
There was one other thing I wanted to ask you about. Do I remember correctly that you were also seeing that some of the people in the trial, since you're taking these blood samples and spinal fluid samples and things, some people who were vulnerable perhaps already had early signs of some of the molecules in the brain that we associate with Alzheimer's disease? And so maybe there's also the opportunity of making sure that now that we have these amazing biomarkers that we can actually look at blood and spinal fluid and detect some of these neurodegenerative disorders before they start expressing themselves in the clinic, maybe that helps us make decisions about who really should undergo these surgeries or whether they need some kind of preparation or some other way of increasing their resilience before they go in there.
Yes, no, you're correct. We do have markers ahead of surgery that indicate that some of our patients have conditions that we typically call pathological amyloidosis of the brain, which has been linked to Alzheimer. So in many ways, there is a real chance that we can study patients who have a predisposition for cognitive decline, including Alzheimer's disease. And if you think about the long-term perspective, we would love to study these patients lifelong. We may actually be able to learn how surgery influenced or may have accelerated or aggravated the condition. So I think there is a direct link to chronic cognitive diseases, including Alzheimer's disease. And yes, to your particular question, we have markers in blood that indicate that some of these patients have a predisposition to develop Alzheimer's disease.
So we were talking a moment ago about things we might do to increase resilience in patients and things that might have broader implications beyond just surgery. Earlier this year, you and your team published some findings that spoke to one angle on that. So Tony Weisskore, who's I think a collaborator of yours, who's been on the show several times and is the director of the Night Initiative. So he famously showed that there are molecules in the blood plasma of young animals that can have a rejuvenating effect on older animals. Of course, this caught some public attention. It was parodied on Silicon Valley with older people taking the blood of the young to stay young. But it's really an exciting idea, in fact, that somewhere in our blood, there is this sort of molecular fountain of youth. I mean, that's an overblown term, but there's something in the blood that actually can affect the aging process. I know that Tony's lab is interested in distilling what exactly is that, what are those molecules. But in your study, you applied this to your interest in the resilience to surgery. Can you tell us what you did there?
Yeah, happy to talk about that. So Tony calls all the proteins in plasma and blood the communicome. And the idea is proteins do communicate with cells. Proteins have signaling functions, and we have many thousands that circulate in the bloodstream. So the idea, could some of these proteins in a higher content and in a certain composition have rejuvenating effect, is sort of the idea that comes out of his work. And one of the issues was, how do we translate that into humans? So in many of these animal studies, you can do a lot of invasive things, and you can not do that in humans. For example, you can take a brain biopsy. You can take the brain out and slice it and die. It's not going to happen in a human being. So is there a way to actually do a study that sort of proves the principle? You give young plasma to an elderly population and you use a stress model to test, does that actually do anything. And if I say do anything, this is really do anything at the level of the biological level. So you would measure inflammatory mediators, you would measure immune cells and immune cell function before and after the fact.
And so this is where your surgical model came in.
And so the surgical model came in. And again, the surgical model, it's an injury model. If you think about injury is, from an evolutionary point, humans had to survive injury. Otherwise, they would not have evolved. So it's a very fundamental challenge that actually activates systems that are very old evolutionary, and the program that's very old evolutionary. And then you respond. The idea is you heal or you don't. And so if you look at surgery in this context of fundamental injury, tests very fundamental mechanisms and differences in how these mechanisms are engaged. So this was the idea. So we have a surgical injury model, and one of the advantages is you can actually do things ahead of surgery, and then you can study things ahead of surgery. You can study things as they evolve during surgery and then after surgery. So the fundamental idea is pretty simple. So you have patients undergoing, in this particular context, knee and hip replacement surgeries, and you would randomize them to either get young plasma or a placebo, normal saline, and you would measure inflammatory molecules, proteins, actually a large array of proteins, as well as immune cell functions before surgery, after surgery. The idea being, does the immune response and inflammatory response to surgery differ in patients who received young plasma, and we're talking 35 years old on average, or normal saline? And so it was a really compelling idea, because is what we see in mice somehow true in humans? So we did the study. We randomized patients to young plasma and randomized them to normal saline. And we were all positively surprised that if you actually give young plasma to patients, you change their immune response and their inflammatory response to the surgical insult. And the way you change it makes sense. You actually modulate pro-inflammatory pathways, and if anything, you sort of dampen the pro-inflammatory response. Interestingly enough, patients who were particularly inclined to be pro-inflammers showed the most profound effects.
Oh, interesting.
Going back to this idea, not everybody is the same. And if you give young plasma to a vulnerable population, they likely profit. If you give it to a resilient population, they probably don't.
Right. Maybe the resilient population already has a youthful immune system.
Exactly, yeah. That was the conclusion of the study, and we were positively surprised that, yes, it's sort of nice, right? What you observe in mice translates to humans. I mean, that's
very nice. It doesn't always happen.
Yeah.
Well, we're almost out of time for today. This has been such a fascinating conversation. I wonder if you could leave us with, where do you see this going? What would you like to see us be able to do in understanding the nature of resilient healing? Yeah, so a couple of things that I already mentioned. One is, can we actually predict who does how? And if so, could we be better consultants to our patients in what a surgical insult actually may mean? And we talk about brain health in this particular context, and obviously the brain matters greatly, but we can think of human health, cardiac health, liver health, kidney health. I mean, there is really broad spectrum. And if you think about resilience, there are many ways to define it and to think about it. But given the 90 initiative, the focus clearly resides with the brain here. So could we make predictions? And if we can make predictions, could we actually consult our patients and make pathway decisions? Okay, maybe not major surgery, maybe minor surgery, maybe conservative approach as opposed to an invasive approach. And I very much see this as a dialogue with patients, because we want to just give them information that they need to make a decision. Somebody who is vulnerable may still decide to have an invasive procedure, but these patients will be better informed. The other pocket, obviously, is what are you going to do about it? Can we actually change trajectories? Young human plasma, it's a little futuristic right down the line. Could we think of a plasma fraction that could be given? Potentially, but there are other things that are maybe more immediate and more practical. If we are able to demonstrate in larger samples using large-scale proteomics, where we measure thousands of proteins, and we can actually start thinking about biological pathways that get activated differently in patients who are resilient or patients who are vulnerable, one possible outcome is that we learn that hyperinflammation is a really
big problem. And hyperinflammers are the one that basically have more inflamed brains and the ones that suffer more severe consequences. So then the logical choice would be, can we actually dampen that pro-inflammatory response? And if you want to do this, how would you do that? And I just give you one snippet that's non-traditional. We can always talk about drugs and medication and pharmacology. But could you, for example, say, we just decrease stress level ahead of surgery? We actually, because stress is associated, acute stress and chronic stress, particularly with the pro-inflammatory profile. So I'm not going to propose this. Maybe I'll leave with a little bit of humor here, sort of the pre-surgical vacation. Could that be a thing?
That sounds like a great idea. Take the vacation, come into surgery feeling as relaxed as you can.
Yeah.
I mean, I imagine there are other things too. I mean, diet influences inflammation. Exercise influences inflammation.
Exercise, absolutely. Dietary interventions, obviously still a young field, but the microbiome, what's the influence there? Obviously, there are many ways, pharmacological, non-pharmacological, interventional, dietary, behavioral interventions that we can think of. And that has become a big topic. The term that's used in the literature is prehabilitation. So you actually get ready before the fact and not after the fact. And that's a topic of significant interest. And again, anesthesia as a field is heavily involved in thinking about these things.
Wonderful. Well, Martin, thank you so much for coming on the show and telling us all about this research in progress. I look forward to hearing more about it as the project goes forward.
Well, thank you. I'm happy to come back and give an update.
I hope you will.
Thanks so much.
Thanks again to our guest, Dr. Martin Angst. He's a professor in the Department of Anesthesiology at Stanford Medicine. To learn more about his research and his project with the Knight Initiative for Brain Resilience, check out the links in our show notes. We actually have a feature article that's just come out highlighting this project. We'll also include links to some of the studies we discussed on the show. We would love to hear from you. Please send us a note at neuronspodcast@stanford.edu with any thoughts you have on the show, your suggestions for future topics in the frontiers of neuroscience, or questions for past or future guests. From Our Neurons to Yours is produced by Michael Osborne at 14th Street Studios with sound design by Morgan Honecker. Our logo is by Amy Garza. The show is hosted by me, Nicholas Weiler, at Stanford's Wu Tsai Neurosciences Institute and supported in part by the Knight Initiative for Brain Resilience. Until next time.